Okay, then I think they are having network issue. Let me just take the citation from my end here. Okay, so permit me to introduce myself. I am Adeokun Said, the current team lead of Ibadan Bacteriophage, and I will be serving as the host for our day two session. And I will be giving a citation of our first speaker for today, Dr. Lavisha Parab. Dr. Lavisha Parab is a postdoctoral researcher at Ludwig Maximilians University, LMU, Munich, working in the research group of Professor Dr. Caroline Wendling. Our research focus explores the evolutionary dynamics of prophage maintenance in Escherichia coli, especially in interactions within microbiome contexts. Our expertise is in combination of experimental evolution, molecular biology, and bioinformatics to investigate fundamental aspects of phage bacterial coevolution. Dr. Parab earned a PhD from the Max Planck Institute for Evolutionary Biology, where she investigated how antibiotics exposure influences lytic phages resistance evolution. Her research contributes to a deeper understanding of microbial ecology and the evolution shaping host-phage relationships. Beyond the lab, she is deeply committed to science communication and mentorship of early career researchers, fostering the next generation of microbiologists and phage scientists. My fellow phage enthusiasts, I want us to welcome our first speaker for today, Dr. Lavisha Parab.
Thanks. I'll try to share my screen.
Your mic is muted, ma.
Is it? Hello?
You are still muted.
But someone just said
Yes,
that
ma.
they can hear me.
Dr. Lavisha, you are still muted. We can't hear you.
Yeah. Someone else can hear me.
Sorry, can anyone hear? I can't hear from my end. Oh, okay. I think it's from me. Let me leave and join back. You can go on, ma.
Maybe I'll turn off my video and then it's better for everybody. Wait, I'm gonna text on the thing. What do you think about that?
Hello?
Hi.
Okay, yeah. You are audible now, ma. Thank you, ma. So you can go on, ma. You can share your screen.
Oh, I actually, sorry. I got kicked out. Sorry. I um share this window. Do you see something,
Okay,
guys?
well, we can see it. Yes, we can.
And now do you see the presentation?
Yes, we can.
And does it move? Does it move?
Yes,
Okay, great.
yes.
Thank you. Yeah, it's because I can't actually see you now, so I have no idea of if it's working or not. But I'm gonna start. So, hi, good afternoon, and thank you for having me. I'm Lavisha, and today I'll tell you how bacteria evolve resistance to phages, and then what side effects occur in the bacteria as a result, and finally, how we can exploit these side effects to our advantage. Sorry. Um, but before that, my journey into science. So I thought that I would structure this more as a research talk as well as a career talk, because I see that there are many early career researchers here. So many of you might have seen this map. It shows the predicted deaths because of antimicrobial resistance in 2050. And what they did here is that they mathematically modeled the number of bacterial infections that would occur in 2050. And then they predicted, okay, what happens? How many people will die if all of the infections are antimicrobial resistant? And in this map, the worse the deaths, the worse the colors. So the stronger the colors, the more the deaths. So especially in Africa and Asia. For example, these two continents might have each above 4 million deaths. And right in there is Mumbai. I spent my childhood majorly in a slum with not much sanitation access. And the spread of these infectious diseases was really a problem. So when I was selected to attend IIT Kharagpur for university, I was so inspired to get into research. Especially this was one of the few universities at the time where you could, actually do research independently, had a lot of labs. And so I tried to get into antibiotic resistance research. And after exploring tuberculosis and genetics here, I first went to Montreal briefly to work on Helicobacter pylori and its secretion system. Now, one of the alternatives of antibiotics is bacteriophages, which then took me to the Max Planck Institute in Plön for an internship. And here I felt so challenged and stimulated and yet intellectually free that I also stayed on for my PhD on phage resistance evolution. And now I am continuing phage research in Munich, still in Germany, as a postdoc. So what are phages? And at this, since I'm starting now and I'm kind of going nonstop and I can't see you, so feel free to say something and interrupt me if there is a problem or you don't understand something. Now, so what are phages? Bacteriophages are viruses that infect and kill bacteria and hence can treat bacterial infections. But we have a problem. Similar to antibiotics, bacteria can evolve resistance to phages too. So like we have antibiotic resistance now, we could have in the future antibiotic and phage resistance. What will we do then? So to prevent this is where evolution research can be an advantage. But how? So to infect bacteria, phages recognize and attach to specific receptors on the bacterial cell. Mutations that modify this receptor cause phage to not be able to attach to the bacteria, and so bacteria are now phage resistant. What's interesting is the side effects. Since these receptors are all over the membrane, such a big change can have side effects. With receptor loss can affect substrate uptake, virulence, their growth, their host adaptation, and especially can also cause a decrease in antibiotic resistance. So you see, bacteria that gain phage resistance lose antibiotic resistance. And so if we combine this phage and this antibiotic in a combination, it can act as a trap, an evolutionary trap. But of course, to exploit these side effects in detail, we need to study bacterial evolution more, which is the overall focus of my research. Today, I will tell you two stories. First, I investigated the mechanisms of phage resistance evolution and hence the side effects on bacteria, including antibiotic resistance. Next, I tested how to exploit this evolution information to make better treatments, that is, design better treatments against microbes by combining phages and antibiotics. After telling you these stories, I will take a small break for clarification questions, just to make sure we're all on the same page. Now, the second story brings microbial communities into the pictures, or say a microbiome. Here I explored how the same thing, that is phage resistance evolution, can change in the presence of bacterial communities, since the evolutionary forces are now different. This work isn't published yet, so I will just give you a sneak peek, but I included it because it's more of a career paths section. And at the end, I will also give you a sneak peek into my current research. Okay, let's go. First, the model system. In our system, phage Phyx174 infects E. coli C using lipopolysaccharides, that is LPS, on the bacterial membrane. Now, Phyx174 itself is a very well-studied phage, and so we had all the tools necessary to study the evolution of it. I don't know if you know, but this was the first phage that was sequenced by, it was the first sequence from the Sanger sequencing era. And E. coli C evolves resistance against the phage by modifying this LPS. And we use the system with LPS as it is very commonly used by phages as a receptor, and bacteria often always, like all the different phages, bacteria can evolve resistance to them using LPS modifications. And again, the genetic basis of LPS structures is very well studied. So let's look at the LPS. The wild type LPS has a lipid A, an inner core here in pink, and an outer core here in green. So what we first did is that we isolated 33 phage-resistant mutants. Like, not artificially, they're naturally evolved and then selected for phage resistance. And what you can see is that almost all of their mutations are in genes that assemble or synthesize the LPS. And based on which gene was mutated, we predicted the LPS structure. And here you can see the variety in the LPS lengths that we could then obtain. If you compare the left and the right, the left are modified in their green part, that is outer core, and the right ones are modified in the pink part, that is the inner core. And so they are called rough mutants and deep rough mutants, respectively. Now, this vast collection of mutants, we can ask interesting questions like, how do LPS modifications affect antibiotic susceptibility? So, we measured the growth of these mutants and wild type in two antibiotics, chloramphenicol and gentamicin, as shown in this heat map. Now, to keep this presentation short, I will just explain the susceptibility. That is the result of this experiment, but I'm happy to go back to this figure during the questions. So first, chloramphenicol susceptibility. The wild type and the mutants on the x-axis here are grouped by their LPS phenotypes. So where the wild type is here, and as you go right, the LPS gets further away phenotypically than the wild type. And the y-axis shows the antibiotic resistance relative to the wild type. So the wild type here is at 1, and when the bars go up, the resistance went up. When the bars go down, the resistance went down. Where there are no bars, it means there was no change compared to the wild type. Now, first, what you can see here is that chloramphenicol resistance does indeed depend on the LPS length. Deep rough mutants that are phage resistant are less resistant than rough mutants. And second is that they're even killed at half the MIC, that is a sub-inhibitory concentration. Second, gentamicin. Here you can see that the resistance is roughly similar to the wild type, and only this kind of group with the most cutoff LPS is less resistant than the wild type, but the difference is not as pronounced in chloramphenicol. So LPS modifications largely don't change gentamicin susceptibility. Okay, but why do we care about that? If you think about it, our objective is to kill the bacterial population. But if we add the phages, phage-resistant mutants will always arise. But now we know that the evolution of phage resistance leads to reduced chloramphenicol resistance. Can we exploit this by combining them? That is, can the presence of antibiotic in a phage treatment reduce the evolution of phage resistance? What I mean exactly is that if we take the resistance in the phage treatment only, so phage only treatment as the baseline, chloramphenicol and phage together should have reduced rate of resistance. And the reason is that in the default phage only treatment, we expect all types of phage resistant mutants to survive. But in the chloramphenicol, we would only expect rough mutants. Now, as a control, we expect a smaller effect for gentamicin, and we expect all types of mutants to survive. So what we did is that we quantified our hypothesis and went back to the lab to test it. What we did is we used a fluctuation experiment, which is a standard way of measuring resistant rates and isolating resistant mutants by using 50 replicates per condition. So what did we find? This graph shows the rates of resistance relative to the wild type, relative to the phage only treatment. So what you can see is that the expected rates are in red and the observed rates are in black. What you can see is that our experiments are in line with our predictions. Chloramphenicol reduces phage resistance up to 5 times lower than the phage-only treatment. And the effect of chloramphenicol is also more than gentamicin. More exciting is that the resistance rates are even lower than what we initially expected. Okay, but is it a coincidence, or are phage resistance rates really related to LPS types? Yes. So here I show how antibiotics influence which mutants survive. So what you can see is on the y-axis is the proportion of resistant mutants isolated of each LPS types. And the types of LPS are on the x-axis. So how to think about this experiment is that our initial hypothesis was, okay, the reason the resistance rates are low is because chloramphenicol will not let deep rough mutants survive. And I can show you here is the recap of those results, which were the basis of our hypothesis. And since we actually got the reduced resistance rates, we wouldn't know if it was a fluke or not, or if we, just got lucky, unless we actually investigate whether mechanistically this the hypothesis was actually the reason. So what you can see as in the default treatment, that is the phage-only treatment, a majority of the mutants, that is 80%, are deep rough, 17% are rough, and a few undetermined. And now, when you add antibiotic, that is chloramphenicol, the trend shifts towards a rough LPS first a little, and then with a little more chloramphenicol completely. So at this concentration, which is basically this line, we found 0 deep rough mutants in our experiment. In our control, you can see the trend shifts only a little. So to conclude, chloramphenicol reduces the evolution of phage resistance, and the reason is that it drives it towards a rough LPS phenotype. But it doesn't work with every antibiotic, so it is important to know about the mechanism in order to know which ones to work. And if you see in the clinical sense, this can be used in a way to know, okay, this patient has this pathogen, it is resistant to this antibiotic and has a phage that is susceptible to. Let's see which phage antibiotic combination leads to the best evolutionary outcome, and then use that to have a better success in phage therapy treatment. Okay, but now that we know the mechanism, can we actually check? I mean, this is a proof of concept, so can we actually check for other antibiotics? Yes, we did those tests, but that is a story for another time. But the main conclusion is that it is possible and even beneficial to study bacterial evolution and then make evolutionarily informed phage antibiotic combination treatments. And I can pause right now to take a few questions, and the rest I can do after the other part.
Okay, thank you, Dr. Lavisha, for the first segment of your presentation. And I believe we all on the call has gotten a whole lot of knowledge on the progress of antibiotics and phage combination again during phage resistance. So, like our presenter said, do anybody at this point have any question she would like to attend to?
Yeah, you can also type them in the chat.
Okay, yeah, you can also okay. Um,
Uh,
I'm not
yeah.
sure if has a question.
Yeah, thank you, Dr. Lavisha, for that wonderful presentation. So my question is, how is the combination of antibiotics and phage cocktail or phage preparation being used in the clinical settings, as in been in clinical trial? Is it how the possibility? Is it still at the experimental level or is being used clinically now? Thank you.
Which part is being used clinically? Phage cocktails or phage antibiotic mixtures combinations? Just to clarify.
Sorry, can you come again? I didn't get that.
Uh, I was asking if your question is about phage cocktails or phage antibiotic combinations.
Phage antibiotics combination.
Yeah, so phage antibiotic combinations are actually used in the clinic, and especially, for example, if you check Benjamin Chan, Paul Turner's works, they also, I mean, logically, it's like, you know, because usually patients are treated with phages after they have been treated for antibiotics for a long time and antibiotics are no longer working. But it would be extremely, I think, unethical to just say, okay, we are now stopping the antibiotic and moving on. And so usually they will treat them with some antibiotic or the other. And there are also some papers where what they did is that initially they had the patient that was not susceptible, like the patient's pathogen was not susceptible to any antibiotic. So they treated them with one antibiotic, that the last antibiotic they had treated them with, and the phage. And then after, I think, 7 days or so, that what they saw is that the remaining pathogens that were still in the system were now susceptible to a different antibiotic. And what they did is then they added this antibiotic in the combination, and then the patient dramatically improved in their outcome. And I think this is where it comes that in that we wanted to do the study so that the doctors don't have to always, or the clinicians don't have to always keep testing which antibiotics to use now, or we could just kind of beforehand know what antibiotic could be used in combination. And so I proposed like a 24-hour experiment. What we could do is that take the pathogen from the patient, isolate it, test it against the different phages, and once we know, okay, this phage is working, what we could also do is then take the phage-resistant mutants and then test them already. And we, instead of maybe taking 7 days to then administer a different phage antibiotic combination, you could already do it from the first day on or the second day onwards. I hope that answers your question.
Yes, Dr. Lavisha, thank you.
Yeah, you can all just call me Lavisha. No need of saying Dr. Lavisha. And there's another question in the comments. I can answer that and then move on to the next part. So unless anybody else has a question, you can raise your hand for that. The Africa Phage Forum APF asks, are these the findings of a preclinical or clinical study? No, the hypothesis was generated on the basis of observations in the clinic, as I said before. I mean, as I said just now in the response to Matoso. But we had, but these are proof of concept studies, so that the doctors in the future or the clinicians could benefit from knowing how evolution-informed treatments can be also applied to phage treatments. And I believe there are other researchers. I mean, we are not the only person to do this. There are other researchers also applying this now in the clinic. For example, I think Jeremy Barr's group is also doing that. In Belgium, I think Sabrina presented yesterday, and she, in their group also, there are other papers where they are now using, where they are also using this in the clinic. So if there are no more questions, I shall move on. Share this window. Send. Continue to share. Do you see the new slide now? I'm going to ask you again. Do you see the new slide?
Yes, Ma, we can.
Okay, great. I'll now tell you about the work that's culminated with me working with my current supervisor, and it is more along the lines of career. So for this project, I collaborated with Mike McDonald at Monash University. Whoops. Sorry. I will talk about the career part a little later. Now, bacteria and phages are not usually present in isolation. I mean, in the lab, of course, we do isolation experiments where, you'll take one E. coli host and one phage, and then you track what is happening with these. Even if it's done in mice, maybe you still continue with one. For example, what can be done in the mice preclinical experiments is that they might take gnotobiotic mice, which and then you inject your pathogen, and then you inject the phage in different ways, and then track survival over the next few days. But bacteria are not usually present in isolation in the nature, but rather there are always other species. And those species can impose then different evolutionary forces. And this can again change phage resistance evolution. And the clinical application of this is that we would know how the evolution of phages will occur, whether, for example, phages can now evolve to infect the phages that are administered as a treatment can now evolve to infect other good microbiome species, and then maybe that is a problem. And so so we looked in this next project into how a presence of a community of the multi-species bacteria can influence the evolution of resistance. Essentially, so we had the same, that we had phages, and we combined, they had one of the bacterial hosts, and we combined them with other species, and we looked at how the resistance evolves over 24 hours. Here, what we wanted to look at is, for example, the bacterial host, here E. coli, can have a different growth rate or fitness compared to other species. And as we saw before, there would be a cost of the phage resistance. And in the previous project, I looked at the cost of phage resistance with respect to antibiotics. But even generally, there are phage resistance costs. For example, resistant bacteria might grow slower. And so we wanted to look whether this would then change or reduce phage resistance evolution. Now, we had two pairs to test. Okay, so we don't want to, again, we want to eliminate that these are only coincidences or flukes. And so we tested E. coli BL21 with a T4 phage, and we tested a different host, E. coli MG1655, with a T5 phage. And what this makes sure is that these two phages have different mechanisms of action, they have a different receptor, and this allows us to estimate, okay, is this receptor specific, is this phage specific, is this host specific, or related to community itself. And we also tested two different types of evolved phages, but that's not so relevant here. What we did is, for example, we didn't want to just test phages, but also recently other scientists have tried whether training the phages or adapting the phages to the host can make it better in an actual treatment scenario. Maybe there again, you have less phage resistance evolution. So I will show you the results now. What you see is what happens, what you see here on the graph is the proportion of the different bacterial species. So E. coli, which is the host of the phages, and these other species that were present in the defined community we used. When we began the experiment at time 0, we had 50% of the population being from E. coli, and the rest only in the other 50%. And E. coli roughly grows a little better than the others, so it should roughly stay the same. So, and these proportions here are in a log graph. So what you see is at the beginning, even though E. coli is about 50% of the community, 24, and it doesn't change much after if you don't add the phages, but 24 hours after, the community has completely readjusted, and basically you don't see any E. coli at all. And so of course we looked at now a log graph, and what we see is that the E. coli is reduced from 0.1% to 1% of the population. And when we did a control with these experiments in monoculture, so when you had single bacteria and single phage, the resistant mutants were always reaching to very high levels in the concentration, similar to the ancestor. And what do you see in the next community? So now this is the next combination, which is the MG6055 and T5. And you roughly see a similar result. But this phage, this resistance is much better at overcoming this community. So what you see is that if you use the ancestral phage, the E. coli still recovers to about 20% of the population. But in the evolved phages, there's not much. And so what you could think about is, why are these different, or what is the key differences between those? And on further research, as I said, this is unpublished research, and so I'm only giving you a sneak peek, but happy to answer questions. So we looked further, okay, what are the reasons behind these differences? And what we saw is that in the LPS resistance. Resistance can occur, I mean, quite frequently. However, it is less fit. So the resistant mutants are less fit. And so they don't actually take over the community, and they stay within 1% or even lower of as a proportion. However, in FHUA, the resistance occurs less frequently, so the number of available mutations is fewer. However, the resistance cost is less. And so they can then rise in the community. So perhaps, now in 24 hours, they have only risen to 20% of the population, but in the future, they could, for example, take over the population, and then we have phage resistance again. So the takeaways, for example, for this in the clinic would be to check which receptor the phage is using and whether specific members of the community, for example, if you are using them in a lung microbiome, then maybe some species in the lung microbiome or in the gut microbiome could also help reduce the resistance evolution of the phages. And this could be, for example, accomplished by testing which niche or which resources the specific species are using. Now, coming towards the career perspective. So in the middle of my PhD, I was getting curious about, how do phages influence microbiomes, and wanted to move towards this direction during my postdoc. And I was contacting other researchers like, can I work with you? What research are you doing? I don't have all the skills, but I am willing to learn. And I met one of the phage researchers at this conference, and she was working in Australia, and she was leaving a lab, but there was a research project that was still going on that I could have joined. And then I was like, I could spend 3 months in Australia. This would be nice. Oh no, but where's the funding coming from? And of course, I'm not going to spend my own money to go there. And then I found that there was a grant that I could apply for. And then I was like, I will also get practice of grant writing, because in many countries, you have to compete always for the grants, and there's very less money. And so, it depends. I mean, for some countries have more money than others, but still you need to have your own grants, and it's really good for your future growth in academia if you can get grants. And so I was like, yes, let's write the grant. And I contacted the supervisor, Mike McDonald, and at Monash University in Australia, and he is also there in the department with Jeremy Barn. And I was like, oh my God, I'll meet all these new phage scientists, and I will be in the new department, and I'll learn how the Australians are doing science. And then and so that's how this happened. And I went to Australia, and I mean, because I got the grant, I was very lucky in that sense. And during this time, so of course, I spent 3 months doing these experiments, and then the analysis when I came back. And this is how I was, able to figure out whether I wanted to go into microbiome research or into phage microbiome research for my postdoc or not. And I highly recommend this if there is an option for you to do that. Now, moving on to my current work. So during my PhD, I looked into just lytic phages, which kill the bacteria, but there are other types of phages which can do so much more. And now I am looking at the dynamics of these phages, so temperate phages or prophages in bacterial population, and of course, in a community context. So phages don't always kill their hosts. And temperate phages, what they can do is they can also integrate into their host's genome. And this genomically integrated phage is called a prophage, and the bacterial host is called a lysogen. And these lysogens formed in this way can be stable for many generations. And so the prophages get replicated into the daughter cells. But prophages can still induce in that they will start the lytic cycle when the bacteria will lyse and die. And so there should be no reason why prophages would survive, because bacteria would then always have a risk But to compensate for the risk, phages, I mean, evolutionarily survive, of course. So to compensate for the risks, phages often carry extra genes or beneficial genes, which can then offset the costs that the phage impose or the risks that the phage impose on the bacterial cell. And as well as what they can do is that they can have super infection exclusion, that is, they protect the lysogen from infection by similar prophages. So, for example, they act like an immune system for the bacteria. So if the bacteria is a host for a prophage, it is a lysogen, then it can, I mean, the phages can still absorb to it, but they will never be able to infect the bacteria. And so they provide a sort of infection immunity. But is lysogeny still a better option, or compared to surface resistance? Or how do the costs and benefits of becoming a lysogen compare to those of receptor modifications? And when you consider an evolutionary trajectory, which direction will bacterial populations evolve in? So we investigated these questions in a small evolution experiment. We exposed the bacteria to either of 4 phages and did the daily transfers for 4 days to track lysogeny. And here you can see the proportion of lysogens in the total bacterial population. And what you see is that in this combination of lambda with the host, lambda phage, you see the bacteria are, the lysogens are about 50% of the population, versus the resistant ones are about 50% as well. And similarly in MEP235 phage, so the lysogens are about 70% of the population and the resistant ones are very few. And this is a log scale. So what you see is that in these 2 phages, however, the story is completely different. This in phage HK629, resistant ones are about the majority, 99% of the population, and the lysogens are only about 0.1%. And here the resistant ones are 99% and lysogens are about 5% of the population. And so this raises a question that even though these phages are very similar, so lambda and HK629 are very similar to each other, and they have the same phage receptor and the same attachment site, despite that, there is a difference in the proportion of lysogeny. And so this raises the question that, oh, are surface receptor mutants sometimes fitter than lysogens? Or how are phages that are lysogens even surviving in the population? Now, if you want to think about the clinical aspects of this, you can think that in our gut microbiome, we have a lot of phages that are in there. I mean, there are some lytic phages and some temperate phages that are going around in our gut microbiome. And some of the bacteria are hosts for them, some become lysogens and some are killed. And so knowing this evolutionary dynamics could in the future help us about, for example, modulating the gut microbiome or for fecal microbiome transplants, and how to navigate these dynamics and how to make these treatments better for humans or more effective for humans, especially, I mean, for diseases, digestive diseases, and also for some, for example, there has been a link between the gut microbiome and our mental health. And so, for example, for these treatments. Now, what I'm going to do in the future is I'm basically trying to find out that even though these phages are very similar to each other, why is there such a big difference in the lysogeny versus phage resistance patterns? And I'm going to look at all the different phage parameters that come in and put it into a mathematical model. So what I try to also do is not just do the experiments, but also look at the bioinformatics of it, look at the mutations, and also do mathematical modeling to understand what's actually going on in the system. And this can help us know what's the actual mechanism for it, which makes the results broadly applicable to other systems as well, and not just to the ones that we tested here. So what I will be testing is, okay, at each step of this process, what differences are there between the two phages, and then which differences actually matter for the question. And the question is, okay, if phage-resistant mutants are fitter, why do prophages exist at all? How are they stable, or which prophages are stable in our microbiomes and which ones are not? And yeah, sorry, thank you. And I am happy to take questions.
Okay, thank you, Dr. Lavisha, for the excellent presentation. And I believe I and the other attendees have quite learned a lot as regards the trade-offs of phage and antibiotics in clinical applications and treatment. So please, if we have any questions, we can raise our hands. Although there is already a question in the chat box that you can attend to that has been dropped since. Should I read out the question?
Oh, I can read it out. So Tadese Iguale asked, did you use the same media, that you use a media that's equally suitable for all species? Yes. So it was, so I'm sorry, I didn't include a lot of details because we started already quite late. So since we started 15 minutes late, I still tried to wrap up my presentation. So I didn't tell you all the details, but yes, we did. So Acinetobacter baumannii and uh, Bailey and the other species that we used, they are all, I think, Staph aureus. They all can grow in LB. And it's been previously, I mean, the reason we use this combination of species is that in the previous experiments in the same group, they had seen that they can kind of coexist, but they have a different growth rate and they stabilize at a different proportion. And yeah, we use the same environment which the species are happy to grow in, but always when like in even in the gut microbiome and even in like generally when you make a microbial community based on how they grow and based on their function and based on their utilization, species are never in the same ratio. So you will find there are some species that are always at a lower amount in the community than the others. And for example, in our gut, it's, I think, Enterobacter fecalis. I don't know if in our gut, but in many microbiomes, Enterobacter fecalis, I think, is the one that always is at a high proportion, and then the others are at a lower proportion. Sorry if I'm wrong about that, but there are differences in proportions at which they stabilize.
Okay, thank you very much for the insightful answer. Yeah, thank you. So I want to ask if we have if anyone has any question before we move on. Okay, thank you, Dr. Lavisha, for the inspiring story and your insights on the coevolution of phages. You also delve into the lysogenicity of bacteriophages and our possibility and effectiveness in using phages alongside antibiotics. We are happy and we thank you for accepting our call and giving us your insightful presentation. Thank you very much for the presentation, ma.
Yes, I'm also happy to connect with you on LinkedIn. I'm sorry, forgot the name of the site. Yeah, I'm happy to connect with you all on LinkedIn if you are interested in the microbiome. Bye.
Okay, you can drop the link in the chat session, and we are happy to connect with you too. Okay, thank you. So having this, the display we can see is a presentation from our partner, Northern Biotech, who are into sales of phage DNA isolation kits, and these are kits that we can use to extract purified DNA samples from bacteriophages. And yeah, more information, you can get information from their LinkedIn at Norgren Biotech, or info on their email at info@norgrenbiotech.com. So moving forward, we'll be going to the next speaker, the person of Sir Abuka Eli, and I will be calling on Kende to give the citation of our next speaker. Okay, Kende is not the call. Okay, Kende is off. So Damilola, can you give the citation of our next speaker? You are not audible again, Damilola. I will have to call on Abdul Hakim. Abdul Hakim, are you there?
Yes, I'm here.
Okay, please, can you give the citation of Sir Abuka Eli? Damilola is having issues.
Okay. Good day, everyone. My name is Tulu Abdulabi. I'll be giving a citation of Sir Eli O. Abuka. Sir Eli O. Abuka is a biomedical scientist specializing in medical microbiology with a focus on antimicrobial resistance (AMR) and the potential of bacteriophage to combat drug-resistant infections. He is the founder of the Phage Hunters Training and Research Program (PHTRP) at the Jomo Kenyatta University of Agriculture and Technology, which established East Africa's first course-based undergraduate research experience (KIOS). His initiative provides students with early exposure to meaningful scientific research and mentorship. Beyond his scientific work, Sir Abuka is deeply passionate about leadership development and advancing the next generation of African scientists by helping them build strong research identities and tackle community-related challenges. Through his mentorship and advocacy, he continues to promote accessible hands-on research experience across the continent. Thank you.
Okay, thank you, Hakim. And okay, Sir Abuka Eli, you can
Okay,
go
can
on
you
and
guys hear
share.
me? Hello, can
We
you
can
guys
hear
hear
you.
me?
We can hear you.
Okay, great. So thank you so much, Akim, for the amazing introduction. I think I'd like to make just a quick correction on the same. The Phage Hunters Training and Research Program is it's a it's an NGO, a non-profit, a standalone on its own. So I am a student at Jomo Kenyatta University of Agriculture and Technology. I'm a master's student. That is where I'm undertaking my medical microbiology course. But Phage Hunters is an initiative on its own, trying to build itself up from the ground. So thank you so much again for the opportunity to present here. I have taken a lot of time trying to figure out what am I going to say, because this is it is a tough topic, especially for a young career scientist. I'm always at wars with the definition of an early career scientist, because apparently this is somebody who has done a PhD or so, somebody who is doing their postdoc. Apparently that is what we call an early career scientist. But my thinking is we need to shift that definition so that it includes our undergraduates, because they are actually the early career scientists by virtue of them just starting up their careers in an early stage. So that whole frustration is what partly inspired the dream of Phage Hunters. And in today's presentation, I've structured it in two parts. So in the first part, I'll basically just share with you guys my story, who I am, and how we came about to do Phage Hunters. And then I'll try and address some of the gaps, or just point them out, at least the ones that I have seen and I'm trying to do my small part to address. And then at the end, I'll show you guys what, as an organization, we've been able to do. We are just a year old and some few months, but some amazing work has been done with the team that I am very blessed to lead. So I think before I start, I'd just like to read the room and get to understand. Maybe I can get 2 or 3 people to just share what are they expecting to learn from this session, or at least when you saw the topic, what came to mind? What are some of the things you're hoping to gain from this? Anyone can just feel free to share what they're expecting so that I can read the room as I get into the presentation. Yes, Akim.
Okay, thank you very much, sir. So, my expectation from the topic and from the speaker as well is to give more insight on how we can increase the awareness of phage research and as well phage therapy in Africa. Because, like, at this point in time, the rate at which people are aware of phage therapy as an alternative to antibiotics is very low, very, very low. So, I'm expecting the speaker to put us through from the experience you have from your NGO. I'm expecting you to put us through on how, and yeah, how basically we can increase the awareness among people in Africa here. Thank you.
Okay. Thank you, Akim. Anyone else who wants to add something? Yes, Sahid.
Okay, thank you, sir. My expectation, in addition to Akim's, is I want to gain insight and inspiration on the ease of getting collaboration outside my own specific region to other regions, where I can collaborate with other scientists to gain more knowledge and insights about phage therapy. Yeah.
Okay, so you guys are really looking to understand the how to get this thing moving. So I do hope by the end I'll be able to answer some of your questions. Anyone else? One last person who wants to share what they hope to gain from the session? Okay, I think because of time, I'll just proceed. Like I said, I'll start by sharing with you guys my story of how even I got into phage science, and it's a personal story. I think I should start by mentioning I'm what we call an unconventional scientist. I was listening to other presentations and people are discussing their data sets and what they have done in the lab. But this conversation will be a bit different. You won't see graphs from the lab. Basically, what you're going to be hearing is sort of a voice of a motivational speaker, someone who is trying to call to arms more phage hunters so that we can build an army to fight this challenge that is AMR. So I think I do love this quote a lot, and this is where my interest into AMR sort of began. This was back when I was still an undergraduate student at Daystar University, and we used to learn about AMR in class. So I knew what AMR is and whatnot. But then that was in third year when you have now to do your project work. And in most cases, I think those of us who are here and are young scientists, you can say, especially in low-income countries, it's very hard to get a project that is actually a meaningful project for your undergraduate. So that was where I was at, trying to figure out, okay, how do I even start a project? And I came across, I don't know why, but I was reading the Nobel Peace Prize, whatever, and then I came across the speech of Alexander Fleming when he was winning the Nobel Prize for antibiotics. And he had an amazing warning to it, that a time may come when these drugs will be available. If you read, you can go and find that quote. It's a very long one, but he says, a time is coming when penicillin will be readily available on the shop. And then the ignorant man will go and buy these drugs and expose these microbes to non-lethal quantities, which will then make them resistant. So that was the whole warning he gave. And that was back in 1945. Forward the story, we are at what now people are calling the post-antibiotic era, because when antibiotics came, I think we all know the story of how they were effective, broad spectrum, they would clear everything, and soldiers would, I think there were even some adverts during the World War II that were saying, thank God to penicillin, he will come back home. So there was an assurance that even those who are going to war, they will come back because they won't die from infectious diseases. When I was listening to Dr. Sabrina's presentation, and she mentioned how they're working to ensure phages are also available in Ukraine, where, as she said, war or conflict sort of speeds up the resistance or the spread of drug-resistant pathogens. So you get the picture of how antibiotics became the next big thing. And then, like he said, the ignorant man, he came, we have started misusing antibiotics, and these are the numbers that are becoming shown. Like, I know we have had these numbers the whole of this week. If you've been attending any session on phages, you must have had the 10 million number. Dr. Anani yesterday mentioned this number has now been tweaked. I think they have changed how they are presenting it. It's between 2025 and 2050, we're going to lose 39 million deaths. Actually, it's just the 10 million now divided into the 4 years to get to that number. It's just, but as you can see, the numbers are getting worse and worse. And there's an interesting report that was done by AMR Industry Alliance. And I think that one is also one of the things that pushed our conviction towards starting phage hunters. And it's a call for us to innovate more, because I think the title for that, you can go and Google it, the brain drain that is affecting antibiotic innovation. So apparently, within the last 2 decades, the number of scientists who are publishing or are researching new antibiotics has halved compared to the over 45,000 researchers in cancer research and these other NCDs, because even for the pharmaceutical companies, it is becoming hard. It's a difficult economic model, because for antibiotics, for you to make a profit, if I am a company that is producing antibiotics, for me to make a profit, I need to sell them in mass. But if I produce an antibiotic today and it's shown to be effective, I have to use it in reserve, meaning I can't just sell it out in masses so that I make a profit. I have to reserve it so that I prevent resistance from occurring. So it's when you look at the economic challenge, even the big pharmaceutical companies are like, I don't think antibiotic innovation is any more it's profitable. So a lot of them are moving away from it. So there's a big challenge even for us as phage scientists to figure out, how do we make this case attractive to pharmaceutical companies, to governments, and whatnot. If you attended our webinar when Dr. Toby was speaking, she mentioned something that really blew my mind. The magistral way of producing phages, it forces us to rethink the model of antibiotic innovation, because this time we shouldn't be asking how much money are we going to make from this antibiotic, because we can't, for phages, they have to be personalized to the particular scenario. So you can't really market a personalized cocktail, for example. those of us who are experienced here, please will correct me if I mention something that is wrong. But the idea is the magistral way of phage preparation forces us to think now, how much are we saving if we don't do anything by not getting phages to market? So even when we are presenting the case of fund my phage research project, or you want to approach a funder, your institution or whoever, you need to find a way to position phage as a solution that helps prevent some of the losses that are associated with antibiotic resistance if we don't do anything. So my personal story into phage research, I think I started and then I digressed. I went into a lot of stories. But I am a child of a farmer, and when I finished my undergraduate, so I didn't do any meaningful project for my undergraduate. I ended up doing a systematic review on phage use in systemic infections, which was a very shoddy work. I don't know why, but due to lack of mentorship and whatnot, that is a story for another day. So I didn't do an actual paper, but I knew I wanted to work with AMR. I had read the story about Professor Strathley and how she saved her husband using phages from sewage, and it was amazing. Like it was interesting, but then I didn't know how to turn this interest into an actual career. So I went home and I decided, let me start doing farming. I kept a flock of a few chickens. So, and this chicken, the first round actually did amazingly well, and I made some profits. But then the second round came in, and one morning I come to look at my chicks. They are sick, they are gloomy, they're diarrheaing nonstop. And my parents, being the ones who are experts in the field of chicken farming, they tell me, oh, go and buy this antibiotic. So I buy an antibiotic, I keep giving the chicken, nothing is happening. So I could see already, now my head began clicking that actually this is antibiotic resistance, because I'm buying different antibiotics, all of them are not working, and I ended up losing 30% of my flock, which was a loss that I felt now. Actually, AMR is real. It's not a story that I read in books. It's real. We have people who are making losses on farms. Now, that was my motivation. I was like, let me go back to school so that I can study more and try to figure out how do I get this solution to markets. So I enrolled for a master's medical microbiology. And luckily at the same time, that was in May, Dr. Toby came to Kenya. So I enrolled in my MSc in 2023, December then. Then in May, when I was finishing my coursework, I came across a Phage Friends networking event. So Dr. Toby Najela of Phages for Global Health was coming to Kenya. And this was just a talk. She was coming to present how we need phages. And at the time, the narrative she was selling was why, as developing nations, we need phage banks. Because if you look at previous pandemics, COVID, for example, we waited a lot on the West to develop therapeutics, and then they had to ship them here, which brought a lot of logistic challenges, and people died. So she was trying to shift the narrative and say, we need to build local capacity around phage for us to be able to stem the silent pandemic that is antibiotic resistance. So I left that room with a lot of questions in my head. Are we doing the work of building capacity within Africa, within low and middle income countries? And that question kept on nagging in my head until it gave birth to what is now the Phage Hunters Training and Research Program. A few months later, I go back to the same institution, Kenya Institute of Primate Research. This time, I'm going to learn about phages, but I was not alone. I went back with a team of 31 people. So me as a student, somebody who had a passion for this, we started by just advocating and saying, this is something that we need to prioritize. And I think the story we kept on selling gave us the people, the partners we needed, or the collaborators we needed to help us actualize that training. Instead of me learning alone, phage isolation, I had to learn it with a team of other people. And it was an amazing experience, I would say. If you've not isolated a phage, please try it. You won't regret it. So that affirmed the idea that the virus that could have a lot of application. I won't go a lot into this slide because I know these are things you know. You know, phages are highly specific. They're self-adapting. They co-evolve with bacteria, and they're available everywhere. So when we talk about the phage bank, it means we need all hands on deck to isolate these phages and get them to this phage bank so that whenever we have a crisis, it's easy for us to deploy them where they are needed. So that keeps on affirming the question that we need a call to arms for more people to hunt for these phages. And I loved the talks that have been there. There's a lot of talk around collaboration and building, sort of like, I think Dr. Mayuk is the one who's mentioned around a global phage alliance, so that because AMR it's not a problem for Africa. These viruses, or I mean, these bacteria don't know borders. You can as easily board a plane here after you've carried maybe some salmonella from a handle of a toilet, and you go to a plane, and that's how a drug-resistant infection gets to start somewhere. So these bacteria they don't know borders. It's a global problem. We can't say it is only our problem as Africans. So we have to work together to sort of craft solutions for it. This is a narrative that I really love. Whenever we talk about phages, we talk about them from the perspective of AMR. But phages, I think they actually should have their own course, or even a master's course on phage science or something, because there's a lot that goes into phage science. And I loved the theme and even the topic, the way you put it. You didn't put it as phage therapy, advancing phage therapy. It is phage science, because when you look at CRISPR, it is an enzyme. How should I put it? It's derived from bacteria's. It's actually the bacteria's defense mechanism against invading viruses. In this case, our phages. So that whole technology for CRISPR is what is being used in genome editing. I saw some headlines where people were saying they've used CRISPR to actually remove HIV from a T cell. And that's amazing, cutting edge research. And that's where the rest of the world is. But here in Africa, I'm sorry to say this, but I think majority of the work that's being done, it's around phages. Even that thing that will excite an undergraduate student is saying, you know what, I've actually identified a phage. I've gone to the field and isolated a phage. I've seen some plaques. That's what's exciting our young scientists. Across the oceans, people are getting excited because they've synthetically modified phages, they've synthetically produced phages that can even attack cancer cells. So the conversations that we're having in the different continents, there's a huge divide, and there's a lot of work that needs to be done around getting this research forward. So what are some of the gaps that I have noticed and we are trying to bridge with Phage Hunters Training and Research Program? Number one is around the education and capacity building. Most of the capacity building programs that are around for Phage are one-off trainings. So you come to a lab and then we bring a few people, we train them, and then that's it. Everybody goes on their own. They have to figure out how they will maybe even get their Phage product. So it's not how should I put it? It's not a sustainable model that would say, I'm actually putting in the work to cultivate and make sure this person that I'm training today, they'll end up becoming a Phage scientist. Because you'll get in a class of maybe 100, even the workshop that we did, not all of the students that we worked with, all of them are working on Phage projects, but it's because it was designed that way. It's a one-off training where you come, we train, you get the skills, but then we don't follow up to ensure the skills that we've given, where you're going to practice these skills. And that's why we're saying, can we integrate phage biology within university curriculum? The model that is there is that you'll work through a course, cram a few information that will then replicate in the exam at the end. But can we then shift this so that we say, these students, we give them the challenge and say, Acinetobacter baumannii is an infection that is causing a lot of deaths. It doesn't have, antibiotics are becoming resistant. Can say class of 2025, can you go out to the field and hunt for phages that we can use to tackle these antibiotic Acinetobacter? And that is shifting the conversation, because for once you're telling the student, you're actually going to be involved in a project that can result in a solution that can save a life. Infrastructure, I think this one we can't mention enough, and I hope even at the end of this talk you guys will help me how to figure out this, because as phage hunters we also don't have infrastructure, and this is where collaboration comes in. As a student, all you can do is come up with a proposal. Once you have that proposal, you say, okay, for my objective 1, I need A, B, and C. These things are things I do not have, but I know of lecturer so and so or a certain doctor who has been doing work with this pathogen. Can I approach them and ask them, this is what I want to work on. Are you able to assist me with a few reagents so that I can move forward from this objective 1, so that it's not just an abstract concept, but then you're trying to answer the same research question as a collaborative venture between you and your supervisor. It doesn't have, I know when we talk about collaborations, you're thinking, I need to collaborate with Phage, I need to collaborate with these big companies. Collaboration starts with who is that person next to you who can help you achieve a certain thing. At Phage Hunters, we are trying to build what we call peer mentors. So everybody on the team, there's something that you know that I do not know, and you have to teach me that which you don't know, and I will teach you. And that's how science is supposed to be. It's supposed to be a collaborative venture where we are all going on this journey of discovery. The context of AMR, I think I cannot stress this enough. I have mentioned it. There's a lot of misuse of antibiotics in livestock production, and we have the moral obligation as scientists, because we know phages can work or are working. We have the moral obligation to understand how do we make them accessible to farmers so that we reduce the overuse of antibiotics. I think Dr. Sabrina also had some very interesting talks when she was talking about how phage resistance is reversing antibiotic resistance, making this bacteria susceptible. And that is, imagine if we had now misuse of bacteriophages in livestock and we're sending them out and they're trying to reverse this resistance. Anyways, that is a random thought in my head, but you get the picture that we have the moral obligation as scientists that we know this thing works. We need to do the work of getting it to the masses. And how do we do that? It's working together with whoever can help us achieve the next best thing. You don't have to look at how do I start the clinical trial, but it's asking, okay, this research has reached here, then who can help me move this from here to here? I think I'll take a pause there, and then I'll just like to hear from the room what you guys are hearing before I share with you guys what we have been able to do at Phage Hunters, what has worked for us so far. Anyone who has a question for me? Are the gaps making sense?
I think there are some questions for you in the chat box.
Okay. So, yeah, I see Africa Phage Forum is asking within JQuat, there's a phage expert, Dr. Julia Kelly. So we haven't worked with her directly so far. So JQuat is my school, but I am studying at the Cambridge Graduate School campus. So interacting with her is not as easy compared to if I was actually in the main campus within the school. But we haven't interacted or worked with her collaboratively. The people we have worked with closely are Ivy Mtai and Dr. Atunga from Kipre. So even the workshop that we did with the help of the Kipre team.
So I think there is one more question that needs to be answered.
Okay. Coming to the ongoing, we are already having a number of phage groups in Africa, including IBRIT, Kipre, phage training groups, Phages for Global Health, Center for Phage Biology. What is your survival model for your NGO? I don't know. Maybe he can explain more on what he means by survival. because I think there are different angles I can take with that. But I'd just like to understand, what do you mean by survival model? How we are surviving? Could you please elaborate more?
On Phage Forum, you can unmute the mic and just ask the question elaborately.
Okay, I think I'll proceed to the second part, which probably might answer um, some of the questions he has. So, what is our structure and how are we doing what we are doing in terms of collaborating and getting people to work with us? So we have what we call dockets, which are our arms of operation. The first one is the advocacy and outreach one, and then the journal club, and then the training and research arm. So the advocacy one is probably, I think, our most mature child on the team. They've been doing a lot of webinars and just summarizing some of the research that has been done, because you cannot say you want to do something unless you understand it. So we have to try and raise awareness around it, because we say our mission is to inspire the next generation of scientists to pursue phage science. So we have to raise awareness and show them the potential of what this is. We did an article on phages as the next trillion-dollar idea, which was just a summary of the article done by Forbes. And it's trying to connect the science to young scientists, because in class, I don't know what your experience is in other countries, but I can say for a fact here in Kenya, most of us, I did biomedical science, and most of us who do the biomedical science courses, you can go through the whole 4 years and not have a mention of phages. What I heard about phages in my coursework was just phages are viruses that hunt bacteria, and that was it. No mention of CRISPR, no mention of phage therapy. So there's a whole world that is not known, and that's what the advocacy and outreach team is trying to do, just raising awareness and trying to get who else is interested in sharing their message around their stories, around how phage research has impacted their work. And then we have the Journal Club, which is, this is our mentorship community or our mentorship engine. And here we train students in critical reading, hypothesis generation, scientific writing, and research design through a structured literature analysis. So basically, and this was actually what gave us an identity when we were starting. We would sit down every Thursday until late at night. We are trying to analyze papers in phage research, trying to understand what are the strengths of this paper, what are the gaps within this paper. And these are questions that in most schooling, you're not taught the skills of, I need to read a paper with a critical eye. Actually, it was very interesting. The person who taught us the critical appraisal is a Spaniard, and he was teaching us, telling us, when you're reading a paper, you don't read it trusting what the author has wrote. You read it trying to question, why did they do their research this way? And that was a different shift from how we read papers. And then the training and research docket is because most of our students who go through our journal club, they end up asking questions. They start, for example, the journal club that we had, our students went through a 20-week program, and we tried to structure the program in such a way that they will read more papers around in silico prophage analysis. So they were, at the end of it, they were asking questions like, so within these Salmonella genomes, we have prophages within these genomes. Okay, what are some of the tools that you can use? We exposed them to FASTA. These students went ahead, they used FASTA, they were able to generate the sequences for the prophage region. Some of them, their project, I think at the moment they're working to see how they can identify endolysins within these prophage regions. So we are trying to tell students that You don't need sophisticated lab equipment for you to do meaningful research, because research starts with a question. It's as simple as saying, I'd like to imagine when Felix de Herel and what is the other guy, Frederick Trott, were discovering phages. They asked a simple question of, what if in this invisible liquid, this thing that is killing bacteria, we can actually use it to treat infections? So it was a what if statement, and that is what has brought about this whole century of phage revolution up to CRISPR. And there's a lot of work that is happening around phage host interactions. If you go online and just search phage PhD, you'll get a lot of work is being done around phage host interactions, evolution, what I think the previous speaker before me was mentioning. So our work is to try and tell young scientists that you have the power by the mere fact that you are a scientist. Start reading literature, understand what is there, identify a gap. And then once you've identified this gap, are you able to write a proposal around it? Once you have this proposal, you don't look at it, I need to achieve this big thing. You start by asking a simple question. For my objective one, I need to go to, say, river, whatever, and get my samples from there and then analyze them. So look around you. Who is the scientist around me that can help me get the lab equipment? Who is the scientist around me that can help me sequence my data? I don't know if I'm making sense, but to try and bridge that thing of collaboration, it starts by, even you as a scientist, are you communicating that which you're doing? Because when we started raising awareness, which was where our NGO started, we were just talking about what has been done and trying to tell people that this should be a research priority within African continent. We need to do a lot of work around capacity building. And this capacity building shouldn't be a one-off thing. It needs to be something that is structured in a way that it is sustainable. And that brings me to, I think, some of the key achievements and what we've been able to do. I'll start with, I think our biggest achievement was when we ran a fundraiser and we raised some money, which helped us even run the 10-day workshop. I don't know how many scientists here who are senior scientists have ever run a fundraiser for their individual research. I am a young scientist, and these are things I have had to learn. I'd say the hard way, because I would hear my lecturer would come to class and they would say, I have been able to raise this amount of funds for KEMRI in terms of grants and whatnot. And as a young scientist, for me, what clicked was, as a scientist, I need to be smart around how do I mobilize resources to fund my research. So it starts by even talking about the importance of your research. You expect the government to fund your research, and yet you're doing phage work. You're only chasing for that work to be in a publication. You don't want to talk about it with key stakeholders in government, in whatnot. You're not talking about it with your students, so that even the students can be able to say, phage research is actually something that I can be in. Because if the students are charged to do it, they'll start even what we call clubs. We had a student in our journal club who has gone ahead to start a journal club in the institution, and I'm hoping that they can start that fire in their school so that they can push even the administration to say, we need to find reagents to fund this student project. So it's up to you as a scientist. I think science communication has been a big conversation. Communicate the science that you're doing so that people can know this science needs to be a priority. We've done a lot of advocacy initiatives. I think these numbers are a bit wrong, but as you can see, we've run a lot of panel discussion, and this just starts with us reaching out to a scientist and asking, are you willing to share your story around the work you're doing in phage research? And the phage community is very welcoming, I'd say. I haven't interacted with a researcher who has been just like, no, I don't want to share what I'm doing. Everyone is always welcoming to share what they are doing. Our team has been able to write a systematic review on gonorrhea. That is the journal club team. It was the first project they did to just try and see, is gonorrhea an actual problem that can warrant phages? We have the workshop manuscript under review. Our trainees, like I mentioned, our undergraduates who did the journal club, have been able to do in silico prophage analysis projects, and they are working towards having manuscripts at the end. So this here is what we are trying to build, or are in the process of building, what we call the CE Phages version of Africa, which is just Phage Hunters Training and Research Program. Because when I came across these guys, I wanted to join them, but I realized they are only supporting students abroad. Actually, I think Ibadan, you guys should tell us how you were able to convince them to come to your institution, because that would help a lot of students. And that's what we are trying to do. Every time I look at this figure and see the number of students they've been able to engage from freshmen to sophomores or second years, and the number of phages they have been able to isolate, it just paints the picture of how universities and research institutions can work together to push forward phage science. So I won't go into these details, but essentially what we are saying is, I feel it is a disservice for me as a master's student to be doing a project on isolation, characterization, and sequencing here in Africa, and yet another student, another master's student abroad in Europe or wherever, the project they are doing is on modifying these phages. What that translates in my head is an undergraduate should be the ones driving the phage isolation and characterization and sequencing, because most of the work of phage comes after you have already isolated the phage and you have the genetic sequence. That is when the work for the phage begins, or the work of phage therapy begins. So we are trying to push the isolation and characterization to universities and sort of try to decentralize phage training within African institutions, so that then the breed of scientists that is coming up, they are coming up to pick from, we have these untreated phages, how can we create chimeric phages or synthetic phages to be able to address, to be broad spectrum? Those are the kind of questions I'm looking forward to having our young scientists asking. This is an amazing story which I love personally because it inspired my passion for phage, and she was also one of our major donors when we started up. She believed in the story that we had and what we are trying to build. And I'd advise you guys to go and find her book, The Perfect Predator, and read it. It's an amazing story. There's a quote that she mentions in the story. It took a village of scientists, doctors, and dreamers to save one life. So what it means is, for us to realize phage therapy, for it to be able to impact a patient, it's not going to be done by one person. It's going to take a whole team of people to be able to get it to the patient. So send that cold email to whoever you think is unreachable. Have a proposal. You can't ask somebody to collaborate when you don't have a plan of what you want to do. Have a plan of, this is what I intend to do. For those of us who are students here and maybe you've already done phage isolation, there was a question during the phage networking event. Those of you who have interacted with Dr. Musila, I love the question she asked her students: who is going to continue your work? And it actually forces you to think about, okay, I have done this project, but is this the end of my work? If moving forward if moving forward that work, it's not something you can do, are you able to reach out to a scientist abroad and just have a conversation, tell them, this is what I've been working on, I have seen what you're doing and I'm passionate about it, can we work together to push this science forward? I don't know if I'm making sense or if I'm preaching to somebody, but I hope I am. So our vision is to build a Pan-African phage network that is not just about isolating phages, but actually driving the innovation within the phage science. We are trying to shift the conversation from just isolating phages in Africa to, can we get a product out of this phage? And how is this product going to help me as a student who isolated this phage? I know there is this conversation that you cannot patent a phage that you have isolated, but you can actually patent one that you have modified. So why are we not teaching our students to modify these phages? So that's my question. And can we create a community where students can do this collaboratively? Because it's amazing when you put students together, you know, that sometimes you think they don't know a lot, but then when you start listening to how they are approaching problems, you try to look at it and say, actually, there's something that these students are teaching me. And that is the kind of community we are trying to build with even our training network. It is a network where we are saying, yes, I am your mentor here, but I don't have all the answers. We are literally learning together. And that is what I believe will, if we adopt such a model for training phage research or phage science, we may end up moving it faster than we probably never thought. There are programs that are doing this. I think I can't stress it enough. We have the Phages program and Phage directory that are trying to already build a community where even phages can be shared where they are needed most. I think what Phage directory does is if you are in need of phages, you can email them and then they can help you within their network find out who has phages that can work on your bacteria. So that is the kind of network that will help us push phage science forward. There are a lot of challenges, like I said, but I think this is what I would challenge us to think. Whenever you look at, say, your environment and you say, I have limited infrastructure, I don't have this, I don't have, try to figure out what is a smart way or an innovative way I can do this. You might end up coming up with a whole new methodology, because I feel where we have problems is where opportunities are. Or at least that is how my brain views problems. I see a problem, I see an opportunity. So don't see your constraints as limitations. Look at them as ways to come up with adaptive methodologies and collaborative models whereby you can work together. As phage hunters, we couldn't have been able to do the training alone, the one that happened at Kipre. But because we had the vision and the plan, we approached Kipre and told them, actually, we want to run this, but we don't have the space. And Kipre were kind enough to open their doors to allow students to come there. We went to Technical University of Kenya, the same. We didn't have, most of our trainees didn't have laptops to work on the characterization or the genomic side of the training. So we asked them, can we use your lab? And actually at the time, the schools, the students were on strike, but we actually were able to use those resources despite even the students being on strike. So it just goes to show how much, when you choose to ask and not see a problem as a limitation and say, okay, how can I work around it? You end up breeding a relationship that can outlast the initial thought that you had. Our vision is to transform every classroom. That is to say, every student who gets into a microbiology course, can they at least hunt for a phage? And by hunting for this phage, can they then breed an identity of saying, I want to become a phage scientist because I want to get these products to the market? So this is some of the work that we have been doing. Like I said, this was our pilot journal club. It ran for 20 weeks. They were meeting every Thursday for a conversation just around how phages affect evolution of bacteria. And more so for them, they focused on the horizontal gene transfer, which ended up in them doing projects on prophage analysis using in silico tools. I think our panel discussions on LinkedIn are something you guys have come across a lot. The image, the one I'm pointing on, this was a stakeholder engagement we were having with the National Research Fund, just trying to ask them, we see this as a potential solution to helping us stem the losses of AMR. How are you guys willing to support even the projects that we have? And this is what I would say, even for the scientists that are here and you're senior, talk about the work you're doing with the people who are supposed to fund it. Like, you can't just wait for the grant to come so that you apply. In most cases, even most grants won't prioritize phage research. You have to find a way of how you will tweak it and, write it very well so that it fits the call. So find a way of making it a priority within your communities, because that is how, the more you talk about phage research, the more people who are interested in funding it or even interested in it will come your way. And then this was, I think, our newest kid on the block, where we are just trying to take the message to students that phages are actually the next frontier in biology. Looking at what CRISPR is being able to do, I don't think we should be reading phages as a one single line within the biological sciences. It's going to transform a lot of things in science, so let's get people reading about it. So this was some images from our 10-day workshop, my first phage hunting experience, and I did it with 31 other people. And we were able to isolate 9 phages, although we haven't done sequencing due to funding constraints, but we will get there when the Lord allows. So, but as you can see, even from the image, only a handful of the students were able to actually go down and collect the samples. So if we are able to get people who can scale this up, imagine if we had 20 students in every institution in Nigeria, for example, hunting for phages across different pathogens. How much phages are we able to hunt for in 1 year? And are we able to get them to the market? So that's also a question we are trying to answer. So I think this is where I'll end my presentation for today. I hope I have not bored you, but my hope is to try and shift the way you think around collaboration. You don't need to collaborate with the big people. Just work with who is around you that can help you push the idea forward. It starts with something as simple as sending an email. You can send 20 in a day and you won't get any. And then one day you send 5 and then you get 1 response. But talk to people whom you think they find value in the conversation around trying to fight antibiotic resistance. If you have a skill that you want to learn, reach out to a scientist whom you think can teach you those skills. We'll be rolling out a few training programs in the coming weeks, so please apply to them. I'm sure that the team has been working tirelessly to plan a hands-on phage discovery that's just very immersive. So have a look out for that on our pages. It will be going out soon. If you're a scientist, a senior researcher, you have lab space, you have resources, you want to collaborate with us to create courses around phage science, we are open. We are looking for people who will help us bridge the gap between the science and the student, so that we don't have students who are just cramming information in class, but are actually doing research within their schools and their spaces. We'll be rolling out a membership joining framework. So if you want to be a member, that will also be going out soon. Just have a look out at our website once it goes up, and let's transform curiosity to discovery. You can get in touch with us by our email. I think I will end there. Anyone with a question to try and I hope I did not bore you and I tried to answer some of your expectations. But yeah, thank you so much for giving me the opportunity to just present and talk to everyone today.
Thank you very much, sir, for the very, very insightful presentation and talk. I believe everyone of us enjoyed and met our expectations of the topic and even beyond as regards phage collaboration and the importance of drawing people around us into phage research and phage therapy. And for me, I think I'm going to stick with the quotes you said, per se. Where we have problems is where the opportunities lie. And yeah, so thank you very much. sir, for the presentation once again. And as for the mail or how to reach out to you as regards the mentorship and other training, you say that it will be good if you help us to type how we can follow up in the chat box. So yeah, so thank you. So I want to ask if we have any question. So now, okay, so Musa, if I'm right, I can't see the rest of the name. If you have a question, you can unmute and speak. Musa, do you have a question?
Good morning, all. Good afternoon, sorry. Can you hear me?
Yes, we can.
Yes, we can hear you.
Just a comment. This is a very wonderful presentation, and we do indeed benefit a lot. We shall try to bridge on fight on this by the use of this page advocacy to fight this antimicrobial resistance. Thank you very much for your effort and your presentation, and the knowledge was really impacted. Thank you very much indeed. That is my comment. Thank you very much.
Thank you, Musa.
Okay, thank you very much. Thank you, Musa, and thank you to everyone. I think in the absence of no question, I want to honorably give the vote of thanks for the webinar series, as today is the end of the webinar series. And I want to first and foremost thank every one of us, our speakers, for their insights, lending us their voice, their knowledge, and their experience in research, their field, to come down to us and dialogue as regards the topic assigned to them, and generally as regards phage and phage therapy. And to all our participants, researchers, listeners, and registrants for the all wide world, we thank you for making this our webinar series of this year, the sixth, to be successful and insightful. I want to also thank our sponsor, Nogen Biotech, for their collaboration and adding more sites and insights to this year's webinar series. To the planning committee and our other volunteers working back end behind the scene, I want to also appreciate you all for your contribution and work and sacrifices towards making this webinar series successful. Once again, Mr. Abuka Eli, I want to thank you very much for heeding our call when we reach out to you. Thank you very much, sir. So, yeah, so thank you, everyone. And I hope to meet all of us more and more in our future webinar series that will be coming up from IBRT in the coming months. And I will also advise that we stay glued to our channels for updates. Thank you very much, and I hope to see us all again in the next World Phage Week next year. Thank you and goodbye all. Yeah, we can leave the call now. Thank you.